Good evening everyone. Good evening all my favorite students. So here we are again. Yes, I know you all have just received your examination results, 10th board results, right? And I hope you all have done very good in your exams, and my all the best wishes for your future and for your further studies. And the ones who have not scored properly or could have done better, for them I would just say, don't worry, we have sufficient time, you will do best and you are going to do much better in your further exams. So here we are today, let's start studying. This is Dr. Anisha from Sri Chaitanya Educational Institutions. Welcome you all for our next zoology session. The various types of questions, various types of queries we are going to discuss today. So I welcome you all for our NEET 2025 Bridge course for the aspirants of NEET 2027. So yes, here we are again. I wish all the very best and I wish very much congratulations for the toppers and the ones who have scored really well. And I really hope you do much better in your future exams, in 12th exam as well as in NEET 2027. Okay? If you have not scored enough, if you think you could do better even, there is a chance in 2027 NEET that you could fulfill your dreams. You could be a great doctor by the qualification of these exams. So now here we are again. So let's start studying today. What are we going to study? Today we have a very interesting topic to discuss. Now what is this interesting topic? We have discussed the various types of diseases and the types of health conditions in the last class. Today we are going to discuss about the various types of diagnostic conditions, or we can say the methods by which we diagnose diseases. Yes, this is a very important topic for your exams. For NEET itself, this is a very important topic, and I will tell you something, that there are various topics here which we are going to discuss today that you may not discuss in future or may not be able to see in future in your actual classes. So listen to this class very carefully, because these diagnostic testings as well as diagnostic methods we are going to study today will be in great detail and will be really helpful for your NEET exams. Alright, so let's start studying. Okay everyone, I hope you all have come out of your zone of results. The results are out, it is declared, you have got the results. Now come out of it, take your pen and paper and start studying. Okay, so first here it is the diagnostic methods. What are diagnostic methods we have here? The various types of diagnostic methods we have here is different types of samples we could collect, different type of test we could do. Now what are these exactly? Diagnosis of diseases that can be infectious diseases can be diagnosed, as well as different type of non-infectious diseases can also be diagnosed. For infectious diseases, which all diseases we diagnose by these methods? The diseases by these methods that can be detected are the various types of bacterial or viral and different other types of diseases. Bacterial, viral, protozoal as well as the worm infections. So these are detected by various methods which we are going to discuss today. Other non-infectious diseases, non-communicable diseases like primarily the cancer. Other than that, there can be diabetes, which we are going to discuss how is it detected. So these are the various methods by which we are going to study today. These are the methods of diagnosis. Methods of diagnosis means the methods by which we are going to study the process or the presence or absence of any disease in a human body. So now what are these diagnostic methods? First it is the microscopic examination. Now microscopic examination of various body samples can be taken. So different types of samples are present. What are these samples? These are the body fluids, or these are the secretions from the body. It can be a urine or any other sample of the body. So the person who is affected, from that person individual samples are collected and microscopically observed. What all observations we do, we will be studying it today. So microscopic examination of different samples from the patients. What could be these samples? These samples such as the blood, samples such as the urine, samples such as the feces, all these are utilized for the processing of the microscopic examination. So for microscopic examination of the different type of organisms, we are going to use, these are the important samples. Other than this, there are many other samples present, but these are the basic samples that we should collect for any further investigations. Radiological diagnosis. Now, what is the meaning of radio diagnosis? Radio diagnosis means we utilize different types of radiations, different types of ultrasonic waves for the diagnosis of the diseases. Now, these are the different types of radiological diagnosis there. So, radio diagnosis means utilization of radiations and wave for the diagnosis of diseases. Radiations and waves. It can be different types. For example, it can be X radiations or X-rays. It can be CT scan, which you have to study in your studies in the NEET 2027 syllabus also, as well as MRI. These scans and techniques are there for your syllabus. Okay, so read them carefully, listen to them carefully. Now, X-ray, CT scan, MRI scan, these are called as the radiological method to diagnose any kind of diseases. These are commonly not used for any type of infectious agents, rather they are used mostly for the diagnosis of cancer. Different types of cancers are detected by these different type of diagnosis. And the third one is the molecular diagnosis. Now, what is this molecular diagnosis? Molecular diagnosis is the method in which different type of genetic material, it is either amplified or it is detected as it is formed. When any kind of infective agent is present in a patient's body, it is identified using these molecular diagnostic method. Using this method, the genetic level or the genetic component of the infective agents are diagnosed. So, they are either PCR, ELISA and many more other techniques are there, but for your examination purpose, these are the two most common techniques that you are going to read further in your classes. So, these are the most common diagnostic methods for detection of different type of agents. It can be infective agents, it can be any other non-infective causes of disease such as cancer and other things. So, these are the most important methods. Now, we are going to study each one of them individually. Alright? Okay, so now here everyone, grab your pen and paper and focus. Just focus on the studies. Do not think about the results. Do not chat among yourselves. This is not the time to celebrate, we can say, or this is not the time to cry out, or we can say we have lost our time or this and that. You have sufficient time right now to prepare for your exams. So, do not chat among yourselves. Do not waste your time. Start studying right now. So, first it is the microscopic examination. What are these microscopic examinations? What are these microscopic examination? Microscopic examinations means we observe each and everything very carefully under the microscope. The name itself is suggesting microscopic examination. So, in case of microscopic examination, what we do? In case of microscopic examination, different samples are first taken. Now, what are these samples? As we have first seen it, it is either blood, or we can say the tissue, or we can say urine and stool samples, stool or fecal matter. So, these are the different type of common samples which we collect for further investigations. Now, what we do if we collect blood or any other sample? For collection of blood or after collection of blood, blood is usually observed for different type of RBC, WBC count, differential WBC count, or along with that hemoglobin measurements, and along with that there are RBC count, RBC measurements are seen. So, these are the methods in which different type of WBCs are individually measured. How many types of WBCs are present, whether neutrophils have increased or not, whether monocytes have increased or not, or neutrophils have decreased, different patterns of WBCs are present, ruptured RBCs are there. All these types of arrangements can be visualized under the microscope using blood as a sample. Now, why are they important? For diagnosis of any kind of infection, neutrophils will increase. Any kind of blood cancer, leukemia, will also show abnormal amount of the WBCs, abnormal patterns of the WBCs. So, how does it look like? This is high or low WBC count. So, this blood examination is required for which of the type of diseases? Blood examination is done mostly for different type of infective pathogens, any kind of infections, infective pathogens. Along with that, cancer, specifically the blood cancer. They will show the presence of abnormal WBCs. In high amount, abnormal WBCs will be present. So, high and low WBC count will be present, because of which we can detect the presence or absence of infection or presence or absence of any kind of cancer. Alright. Other than that, we have urine and stool sample. Urine sample for diagnosis of any urinary tract pathogens. It can diagnose the presence of urinary tract infections, or we can say urinary tract pathogens, and the fecal matter or the stool sample indicates the presence or absence of any intestinal pathogen, or we can say intestinal parasites. Presence of any intestinal parasites. Parasites which can be present in human intestine, such as roundworm are there, tapeworm are there, and many other pinworm are there. Many other numerous types of worms are there which are present in the intestine of humans. These pathogens cause different type of infections. It not only stays in the intestine, it spreads throughout the body even. So, for observing them, to identify them, presence of the intestinal pathogens are very necessary in the stool sample. In the stool sample, these pathogens or these intestinal organisms release some of the ovum, or we can say the eggs, or in some other forms they are present. So, these are called as the different type of samples which we commonly use for microscopy. Now, other than these samples, one more very important sample is present, which is called as the tissue sample. You may have heard of it, tissue biopsy. The word biopsy. I am very sure you all have heard of it. How many of you have heard the terminology biopsy? Raise your hands. Okay. Anybody who has heard the term biopsy, or what is the meaning of the word biopsy? Can anybody tell me what is it? Okay, stop talking among yourselves. Who will be the first one to answer about biopsy? What is a biopsy, or have you heard of the terminology biopsy? Okay. Are you guys studying or not? Or you're just chatting among yourselves? You're just talking about your result? Afshin has. Okay. Who else? Pooja has. Akshara has. Very good. So, you have heard the term. Okay. Sushant is hearing it for the first time. Okay. No problem. Okay. No problem. We'll discuss it now. So, what is this terminology called as the biopsy? Now, the whole terminology is called as tissue biopsy, or also called as punch biopsy. What is it? For example, if a person is detected with a different kind of a cancer, the cancer can be at any part of the body. It can be in the oral cavity, stomach, esophagus, lungs, or any other parts of the body. Now, what is it done? Now, where the cancer is suspected, from that part, for example, we have here taken the tissue as lung. If we are suspecting lung cancer, from the lung, through a needle or an aspirate, what we do? we take a small piece of that cancerous tissue, or we suspect them as a cancer tissue, we take or punch it out, take it out in the form of that tissue. We make thin slices of this tissue, we stain it and we observe it under the microscope. That is called as biopsy. So in case of biopsy, whatever tissue we are taking, whatever tissue we suspect it to have a cancerous growth, what we do, we take a pinch of it, we take out of it some part, we make thin slices, we stick it on a slide, we stain it with colors, we stain it different dyes and we observe it under the microscope. This whole process is called as tissue biopsy. Now what we see under a tissue biopsy, here maybe the diagram you might not understand or might not be able to clearly see things, but in a tissue biopsy, what all we can see? In a tissue biopsy, we can see different type of epithelial layers, the abnormality of the epithelial cells, abnormality of the basement layers and many more changes that a cancerous tissue or a cancerous growth will show. The growth of the cancer or a tissue which is multiplying abruptly, which is showing abnormal cellular structure, abnormal growth of these cells, all these abnormalities are directly seen under the microscope by a process called tissue biopsy for cancer suspected specimens. So that is called as tissue biopsy. So now when you are going to study cancer, you are definitely going to see that tissue biopsy is one of the major process which will confirm the presence or absence of cancer in an organ. So this is about the microscopic examination. So in case of microscopic examination, what all we do? In case of microscopic examination, we take blood, urine, feces or stool. These are the samples we take. Along with that, there is another tissue which is called as the tissue biopsy, tissue from a suspected cancerous organ, and from here directly we punch out, we take out a piece of this tissue, we make thin slices, we stain them and we observe under the microscope. So these are the all microscopic methods which we use for different type of diseases which can be communicable or which may not be communicable. They can be non-communicable diseases like cancer also. Now next we are moving on to the part which is called as the radiological diagnosis. So you guys are hearing, we could hear that you are not studying, you are not attentive, we could see that you are chatting among yourselves. So I would suggest all of you to stop doing that immediately. Focus on your studies. This is the time which you should not waste. This is the time you should utilize at a proper way. So here we are again. So now it is the radiological diagnosis. So now what is radiological diagnosis? Okay. So radiological diagnosis means the various radiations or waves utilized in detecting presence or absence of any abnormality inside a tissue. For example, if someone has a broken bone, how are we going to know about it? If someone has a broken bone, we are going to know about it by X-rays, X-radiations. We will pass through it and we could see the broken part present in the arm or in the suspected area. Similarly, if this is not a bone, if a damage is there in an organ, the internal organs will not be clearly visible by X-rays, the X-ray that we are seeing. So what we do to see those damages present in an internal organ? Bone is a hard tissue, alright, you all know bone is a hard tissue. So these hard tissues can easily be visualized under the X-ray radiations. But what happens if it is a soft tissue like a liver, or we can say kidney, or we can say stomach? These are all called as soft tissues. So soft tissues cannot be observed so easily in X-rays. So what we do? For that we utilize different type of radiological diagnosis which is called as CT scan and MRI. Okay, so let's start studying here. Radiological diagnosis first. What all we are going to study in the radiological diagnosis? First it will be X-rays, another is the CT scan, and the third one is MRI scan. X-ray, CT scan, MRI scan. These are the different types of radiological method we use for diagnosing different type of disorders. So first it is the radiological diagnosis of X-rays. So how these X-rays are useful? X-rays, X-radiations, these are a different type of radiations. These radiations, when they pass through a hard tissue, what it does? The hard tissue prevents the pathway of these radiations, whereas the soft tissue allows its movement. So as a result of which we get an image of these hard tissues called as the bones. So most of the hard tissues, the bones, we are capable of observing under X-ray radiations, like different type of breakdown of the bones, damages of the bones, and many other images can be taken regarding the structure of the bone. Remember, this is a two-dimensional image. X-rays or X-radiations produce here, what we see in a photographic film is a two-dimensional image. Alright. Now what is it if we use the same X-rays and we get a three-dimensional image? Yes, if we are using the same X-rays in a different way, maybe in a more we can say sophisticated way, or we can say in a much stronger way. So what will happen? We will observe a three-dimensional image. How? The X-radiations passing through it in a whole throughout the parts of the organ, as well as when it is attached to a computer screen, it is attached to a various types of programming, we get a three-dimensional image, and that is called as the CT scan, computed tomographic scan. Now computed tomographic scan, what is it? The procedure uses a narrow beam of X-rays. CT scan is nothing but the X-rays, but since the X-ray is producing the image in a two-dimensional way in a photographic film, here they are producing three-dimensional image and it is connected to a computer screen. So here the procedure uses a narrow beam of X-rays to pass through the body, and these X-rays are detected by sensors on the other side. You may have seen the CT scan machine where these things are done. The computer analyzes the data from the sensors to create a series of cross-sectional images or slices of the body. Now how these images are produced? For the formation of these images, what we do, when the CT scan, here is the photo of a CT scan machine. This is the table in which the patient lies down, and the patient is taken inside the machine where throughout X-ray beams are allowed to fall on the patient's body. What happens here? As a result of which X-ray radiations will pass through the body of the individual organ of the human being. As a result, we can get a three-dimensional image when it is connected to the machine, when it is connected to the screen of the computer. So computer analyzes the data. The computer will analyze the data, what it is getting, and individually it will make various slices. Thin slices are produced, and these slices individually will give each and every section of the organ that is undergoing the process of analysis. So suppose if we have to observe the lungs as the CT scan. So when we are placing human body inside the CT scan, each and every slice, the lungs will be kept, or lungs will be studied, suppose under 1000 slices, and each and every slices is then read carefully using computer screen and computer analysis, and as a result, the result we will obtain, we will get the result in that way and we can interpret the result. So it is same X-rays or X-radiations we are using for CT scan also. The only difference is that CT scan is allowed to fall, the X-radiations are allowed to fall three-dimensionally to an organ. As a result, each and every part, each and every slice of the organ can be seen using a computer. And when these individual slices of the photographs are allowed to analyze together, it will give the whole image of the organ. So CT scans are used to identify a wide range of conditions. What are these wide range of conditions? During any kind of injuries, infections, tumors and other diseases. Specifically any kind of injury, accidental injury that happened, or any kind of tumor or cancer formation is there, CT scan is a must for that condition. So CT scan utilizes the same X-rays, X-radiations, but in three-dimensional image formation. Now it is the another type of radiodiagnosis which is called as the magnetic resonance imaging. I hope you have learnt about this CT scan, because CT scan and identification of the MRI, they are as important as you are going to study further in your examination of NEET. So health and disease chapter includes the diagnosis of cancer and the various other infective agents also through the CT scan as well as the MRI imaging. Now what is this MRI? Magnetic resonance imaging. What happens here? So this is a machine which is called as magnetic resonance imaging. The name itself suggests magnetic resonance. Now what is this magnetic resonance? It utilizes magnetic field. The magnetic field will be utilized, and as a result of the utilization of the magnetic field, what happens? The magnetic field is allowed to fall at different parts. This is the cut open section, or we can say the cross section of an MRI machine. So here we have the cut parts which contain coils. These are the parts which contain coils which will produce the magnetic field. Now the magnetic field will be produced by these coils. These magnetic field along with the radio frequencies. Radio frequency, yes, the radio frequency will be utilized along with this. The magnetic coils will produce the magnetic field. Utilizing the radio frequency, an image will be produced. So here MRI scan is this. Using a strong magnetic field and radio waves, a detailed image of the organs will be produced. Alright, and it is used to diagnose different type of wide range of condition. Now what is the difference? We have understood the principal difference between CT scan and MRI. CT scan also produces three-dimensional image of an organ, of a structure, and MRI also produces three-dimensional structure of an organ. CT scan produces in different slides. Slides means the whole organ may be divided into 1000 slices. Each and every cross section, each and every slice of that organ will be studied carefully by computer, and then it is analyzed as a result. Whereas in case of MRI, it is not studied as individual slices. What is it seen? It is seen as the complete internal and external organ. The whole organ as it is will be studied using magnetic field and radio waves, as a result of which we obtain an image which will give us the clear view of it. These are the difference in the images of X-ray, CT scan and MRI. See here, X-rays, it is the two-dimensional image. MRI produces three-dimensional image in which soft tissues, bones, everything will be observed. And CT scan produces the three-dimensional view of the X-ray. This is the two-dimensional view of the X-ray, and that is three-dimensional view of the X-ray, which is called as the CT scan. Whereas in case of MRI, we can see all types of tissues, organs, structures, ligaments, cartilage, bone, everything can be seen by MRI. So MRI gives much more detailed image of the individual organ than CT scan. But both have their own individual utilization. CT scan is utilized for different type of cancers and other techniques. Even MRI is also utilized for different type of injuries, trauma and cancers. But the utilization will be decided by the physician himself. Now we are moving on to the next part of the diagnosis, which is called as the molecular diagnosis. Now what are these molecular diagnoses? These molecular diagnoses are the ones in which molecular level changes are there. Before moving on to the molecular level, I am very sure you all must be knowing different types of molecules are present in our cell. Now what are these different types of molecules? These can be molecules like carbohydrate or protein. These can be molecules like amino acids and glucose. So we are talking about nucleic acids in our body. Nucleic acids present in a cell, which is nothing but the DNA and RNA. So human body has specific DNA and RNA. When human body is infected by a bacteria or a virus or a pathogen, or suppose abnormal growth of tumor is taking place, in those conditions what happens? The viral RNA or viral DNA, abnormal tumor DNA, all these will be present in human body, and these will be detected by molecular diagnostic method. That is why the name molecular diagnosis is given. Molecular diagnosis name is given because at the molecular level, or we can say the molecules such as nucleic acid level, we are detecting the presence or absence of disease. That is why it is molecular diagnosis. Now what is the use of the molecular diagnosis? Molecular diagnosis is, first of all, we can say PCR. This is there in your syllabus and you need to study them in detail. Polymerase chain reaction, PCR, polymerase chain reaction, and another one is the ELISA, which you are going to study for your exams also. So PCR and ELISA we are studying here, or we are going to discuss here. There are many more methods which are the molecular methods, but the importance of those are not there as per your syllabus. So that is why we are only restricted to the PCR and ELISA. ELISA stands for enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. Immunosorbent assay. So you all have not stopped talking, right? See, that is the problem. You need to listen to your teachers. Even the chemistry class was also, I saw sir was telling you all to stay quiet, but none of you heard of it. Here also the same thing, but the question is, you are just wasting your time if you are wasting your time in the chat box. I hope you all are listening to this because these are the ones which will help you in the exam. Friends and other things will not be there for you. These examinations will be based on these knowledges, these topics. So you must be listening to this. Okay. So again, the last time I am telling you, listen here, stay focused to your studies, stay focused to the topic that is going on right now. Do not get distracted. Getting distracted is the most easiest thing you can do right now. But staying focused will be difficult, but that will keep you on the track. That will give you the result in your future. So coming back to you again, PCR, polymerase chain reaction, ELISA, enzyme linked immunosorbent assay. Now what is this? Polymerase chain reaction is, polymerase chain reaction in this process, polymerase chain reaction in this process, what is it? In this polymerase chain reaction, a single genome, it could be the presence of a DNA or RNA of an infective pathogen. For example, you have heard of the COVID-19 RT-PCR. I am very sure you belong to the COVID era, you are the kids of COVID era, and so that is why you all must be aware of the terminology RT-PCR, that is RT-PCR. RT here stands for real-time PCR. Even sometimes RT also stands for reverse transcriptase PCR too. So that is another part of it. There are different types of PCR. So first of all, we are coming to PCR. PCR stands for polymerase chain reaction. There is an enzyme DNA polymerase which can multiply a single strand of DNA into multiple times. Using this principle, the polymerase, polymer formation, DNA will be polymerized, multiple copies of DNA will be produced. Using this enzyme, what we do? If the human body is infected by a viral pathogen, the viral pathogen will definitely have either DNA or RNA. So this DNA or RNA present in the body or the blood of a human being will be taken and it will be amplified multiple times, million times using the polymerase chain reaction technique. So suppose maybe 3-4 viral particles are present in the whole body, which will be very difficult to find out in any other method. So these 3-4 viral particles, if present, will be taken up by the blood and may be polymerized by the process of polymerase chain reaction. Multiple copies of it will be formed, so it will be amplified and will be easily visualized for the presence or absence of the disease. So that is called as the polymerase chain reaction. Polymerase chain reaction, it is in the form of a chain reaction. You are going to study each and every step, each and every process of it. But right now, at least remember, polymerase is the enzyme. The different type of DNA polymerases are there. A specific type of DNA polymerase is used over here for the amplification of the genetic material of the pathogen. Now this pathogen can be a virus. We have taken an example of virus here, but it can be a bacterial pathogen also. So a bacterial pathogen, a viral pathogen or a parasitic pathogen, it can be a malaria pathogen. So many type of pathogens can be utilized for the amplification of their genetic material, and these genetic materials are further analyzed for the presence or absence of the disease. Okay. Now other than this, other than the pathogens, the presence or absence of cancer cells, the presence or absence of the cancer cells, that is also done by this same method of polymerase chain reaction. Now in case of polymerase chain reaction, what is it done? In case of these cancer cells, what is it done? Cancer specific components will be present in case of growth of cancer cells. These components might be at the initial stage of cancer at a very low level. So what is it done? It is taken up, these cells are taken up, now they are amplified and observed for the presence of these components in the cell. If these components are present in the cell, it will be called as the presence of the cancer. If these components are absent, cancer is not detected. So presence or absence of cancer, presence or absence of any pathogenic agent in the body can be detected by amplification of the specific genetic material present in the organism or the particle. That is polymerase chain reaction. So this is the unit of polymerase chain reaction. It is not a huge machine, it is a small machine. What we do here, in case of polymerase chain reaction, sample is added to these vials and then further it is processed at different temperatures so that at different level the DNA will be processed and multiple copies of it will be formed. The whole processing definitely will be discussed in your actual classes. Over here, just remember the genetic material of the cell, whether it is a viral or a bacterial or a parasite or even a cancer cell. The genetic material of the cell will be multiplied or amplified million times so that easily their genetic component can be analyzed and the presence or absence of the disease can be detected. So here what we do, we just amplify the genetic material of these cells, and for this process we need this technique called as polymerase chain reaction. So this is the step of polymerase chain reaction in which this is a single copy of DNA. The single copy of DNA will be separated, then they will be attached with primer like structures. You are going to study them in detail later, and see here we have 2 copies of the DNA. So by a single piece of DNA we already got 2 pieces of DNA using polymerase chain reaction first step. Now what happens when we are utilizing this cycle many times, millions of copies of DNA will be formed. So that is how we amplify the genetic material of the DNA, RNA or any of the component of the infected cells. Now it is ELISA. What is this ELISA? ELISA is enzyme linked immunosorbent assay. So it is enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. Alright. We have seen PCR is utilized for various types of infective agents and cancer. Infective agents like HIV, infective agents like COVID-19, infective agents like tuberculosis, and cancers, different types of cancers are detected by PCR, polymerase chain reaction. Now there are different types of infective agents which are also detected by enzyme linked immunosorbent assay. Molecular tests are the last stages of diagnosis. The initial stages are the blood and various other examinations. Other than that, we can go for the radiological diagnosis. Molecular diagnosis is the last stage where we need a confirmatory result. So now it is the ELISA, enzyme linked immunosorbent assay. Now what are these enzyme linked immunosorbent assay? In this process, we take plastic, or we can say micro titer plates. These are called as micro titer plates. You are going to see them later. So these micro titer plates are used for the process of ELISA. In ELISA, we have studied antigen and antibody earlier. So in case of ELISA, antigen antibody interactions are utilized in future. So what are these? So the whole process of ELISA, it is dependent on antigen antibody interaction. These antigen antibody interactions in detail you are going to study for your exams, for the NEET preparation in your actual classes. But right now, just remember antigen, antibody, either of them labeled with an enzyme. Enzyme you all must be knowing, enzyme as well as the substrate. So enzyme as well as the substrate component of it, maybe it will be too difficult for you to understand right now, but I would suggest you to understand that there is component called as the antigen we have studied, there is a component called as antibody we have studied. Now in the body, either the presence of antigen or the presence of antibody is detected by a chemical called enzymes, and enzymes has a specific substrate to it which produces these colored components. These specific colored components are seen. You can see here the whole plate appears yellow colored. These are the solutions which are yellow colored, and some of them are bluish in color. Some of them are blue colored. Now these blue colored components are the ones which is giving the positive result. So there is a very complex mechanism in the ELISA. Okay. So it is a very complex mechanism in the ELISA which you don't have to study now, maybe little bit in the actual classes also. The only thing that you need to remember right now is antigen and antibody. These are the ones which we have studied. Antigen is the pathogen that has entered our body, and antibody is the immune response that our body is producing. So antigen and the antibody, they will be linked with the enzyme named chemicals. These enzyme as well as the substrate chemicals will be linked with any of it, and it will produce a colored component like this, which we are going to see as the result. So at least remember, antigen antibody interaction produces results which appear in the form of colors are the ELISA test. So that is enzyme linked immunosorbent assay. So enzyme linked immunosorbent assay and polymerase chain reactions are called as the various types of molecular diagnosis. So any kind of disease, any kind of infection, we do not directly go for the molecular testing. What we do, we start our test with the preliminary test like the different type of examinations of blood, stool, urine and tissue. And further we go up to the radiological diagnosis, and before or after that we go for molecular diagnosis. So this is how we can diagnose any kind of infections, any kind of diseases. So this is another various type of pathogens or various type of components that are present in each of the well of microtiter plate. So this is more complicated. Right now what you need to understand, antigen antibody reactions in the ELISA test. So that is all for today. I hope you had a great time, you had learned something, and I wish you all further very best for the future exams. I know most of you have studied well and most of you have scored very well in your exams, but those ones who have not scored enough or you think you could do better, don't worry, there are future exams again. Prepare for the NEET 2027 and whole Sri Chaitanya group is there for you. All the best for your future exams. Thank you everyone.